Okay.
Good
Hello
morning.
and welcome. I'm Coleman Kennedy, CEO of the Neuromuscular Disease Foundation, and we thank you for joining us today. As part of our commitment to support the GNE myopathy community, we are pleased to present our esteemed board member, Dr. Tahseen Mozaffar, professor of neurology, pathology, lab medicine, and orthopedic surgery at UC Irvine. He'll be speaking today about emerging therapeutic approaches for GNE myopathy, such as sialic acid-based treatments, along with a brief overview of current gene therapy research. He'll also cover what patients and families should know about clinical trial participation, followed by audience Q&A. If you have questions, please enter them into the chat box anytime during the talk, and we will address them after Dr. Mozaffar's remarks. With no further ado, here he is.
Thank you. Thank you, Coleman. This is wonderful. I have always enjoyed interacting with patients with GNE myopathy and been seeing a lot of GNE myopathy patients from all over the country and most recently from Canada. So this is, I think, a good time to talk about what has happened in GNE myopathy in terms of therapeutics and where we're going from here in terms of both clinical trials as well as possible gene therapy efforts. So I've been at UC Irvine for 26 years and have worked very closely with the NDF over these last 2 decades, and I'm on the board for NDF. I do a lot of clinical trials in all sorts of neuromuscular diseases. My focus is primarily rare and ultra-rare muscle disorders. So these are all my disclosures related to that. So what I hope to achieve so far is one, to give a little bit of a primer on GNE myopathy for those who are relatively new, newly diagnosed, or have not heard one of my previous talks. I'm also a unapologetic history buff. So I would talk about a little bit of the history of GNE myopathy, but then my focus will primarily be on therapeutic trials. So I will cover the trials that have been done in the past, including the extended release sialic acid trial, the MANAC trial, and the more recent Korean trial on 6-silolactose. We will definitely talk about the most recent trial that is about to start, and we will talk a little bit about it. And I know that Dr. Salman Bana, who's a new member of the board, will be talking about gene therapy in the near future, but I just wanted to spend some time talking about what the efforts are. So, GNE myopathy is a relatively new disease in the sense that it was described for the first time in 1981 by Dr. Nonaka in Japan, and he called it distal myopathy with rimmed vacuole, and this was primarily based on the clinical presentation and the pathology information. Now, soon afterwards, Zohar Argov independently described a similar disease in the Iranian Jews living in Israel. And again, he saw the rimmed vacuoles on the muscle pathology. And one of the observations he had was that this myopathy, despite its severity, somehow spared the quadriceps muscle, which is a very important clinical distinction that Zohar found. Now, soon afterwards, the groups got together, realized that what Nonaka had described and what Zohar Argov had described are essentially the same thing, which is they are the same disorder. We just call them by different name. And we also realized that this particular entity is not unique to Iranian Jews. It can actually happen in non-Iranian Jews as well. And I'll show you some data how this may be one of the more common forms of limb-girdle muscular dystrophies. And then in the late 90s, they were able to find the gene responsible for this disease. It's a gene that we had known about because it also causes a childhood metabolic disorder called sialidosis, but it turns out that the same gene can also cause GNE myopathy, depending on which portion of the gene is mutated. So it's known as distal myopathy with rimmed vacuoles. The Japanese, in deference to Dr. Nonaka, still like to call it Nonaka's disease. And we know that it was a little bit more common in Iranian Jews, but we have had more information. So it is a recessive disease, which means it skips generation. The onset is usually in the late teens and early 20s. It presents with bilateral foot drop as the first presentation. Then it progresses to the hamstrings and then ultimately to the hip flexors, sparing the quadriceps muscle in the process. In the Israeli population, there were some cases that presented primarily with hip weakness more than the foot weakness. And then ultimately, it will affect the arms and it can be very severe, but the rate of progression and this degree of severity is variable. And it really depends on the gene mutation and a bunch of other factors that we don't fully understand. So we've had patients who are in their 70s with minimal disease, and then there are patients who are younger with very severe disease and already confined to a wheelchair. The progression is slow. It goes from distal muscles to proximal muscles, and there is no involvement of the heart. The respiratory muscles get very rarely involved. So it's common in Japan. It's common in what's called the Mizrahi Jews, which are Jews that originated initially from Middle East, from Iraq and Iran, who also then went to the Indian subcontinent, but it's also quite common in the South Asian Indian subcontinent. And now the more you look for it, the more you find it. So it's prevalent in Arabian Peninsula. It's prevalent in Japan, in China as well. And in some patients, you may get low platelet counts, which is one diagnostic clue that happens in these patients. So this is just in a nutshell, typical onset, second decade, foot drop progressing to more proximal weakness, spares the quadriceps. It's a recessive pattern, which means that you need to have two separate mutation, and which means that you inherit one from your mother and one from your father. And that again raises the issue of so closely inbred populations where they tend to marry within the same ethnic group, or in families where there is consanguineous marriage, which is cousin marriage. These are relatively common. There's a specific pathology that we see on muscle biopsies. It's a non-inflammatory muscle disease, so there's no need to use steroids or anti-inflammatory medications on this. And this is a recent paper from Madhuri Hegde and the Indian collaborators. And what they found was that if you do genetic screening in patients with limb-girdle muscular dystrophy, GNE myopathy shows up quite frequently. And this is a Indian population. This was a study done in Bombay, but I think they had other centers from India. And even though we typically think of dysferlin, which is DYSF, as the most common form, in fact, in their population, GNE was the most common cause for limb-girdle muscular dystrophy, and happened to be the third most common cause of genetic myopathies. And the same is happening in other literature as well. So even in the Caucasian, it is in the top 10% types of limb-girdle muscular dystrophy that you may find GNE myopathy. So I'm going to skip this, again, just to point that it's mainly a distal weakness with leg weakness. And there is a steady and rapid progression in most patients. So this is looking at the upper extremity composite score on handheld dynamometry, which is one way of testing your muscle strength. This is the score in the lower extremity, and as you can see that over a period of 36 months, there is a very steady decline. And these numbers are important because this is what will help us to calculate the power for a study. So if I want to do a clinical trial, I need to know what the rate of progression is, so I can estimate how many patients I would need to show a meaningful change in the slope. The goal in neurology is always to either stop this progression or significantly slow down the progression. And once you stop and slow down the progression, the muscle has a chance to heal itself, and you may see some improvement in the long run. That takes longer because the muscle has to regenerate. Many years ago, for the Ultragenyx pivotal trial in extended release sialic acid, a functional activity score was also devised, and we know that the functional activity score also declines with time in GNE myopathy. And the 6-minute walk test, which is the traditional measure of endurance in muscle patients, also declines with time. So the defect is in the sialation pathway. So the glucose is ultimately converted to sialic acid. Sialic acid is an important constituent of muscle and muscle membrane, and there are enzymatic steps that take you through that conversion. So you have many critical enzymes like MANAC that was recently tested in GNE myopathy, sialic acid, etc. And ultimately, these glycoproteins are processed in the cell, excreted out, and they're usually expressed on the cell surface. If you don't have sialic acid, you get GNE myopathy, and GNE myopathy presents usually with myopathic features of muscle changes on muscle biopsy. There's often rimmed vacuoles. These are these holes in the muscle which are lined with material. You can see it on this H&E stain, or you can see this on a trichrome stain. And if you do a electron microscopy, this is filled with autophagic material, or junk as we would like to call it. And you can see that on staining with some of the other enzymes that are specific to autophagy, etc. So this is shown here. You can see this, that there is a disruption of the muscle membrane, and if you have a successful treatment, that may potentially change what the muscle looks like. So far, as we have talked about, we have very little in the way of successful treatments. The enzyme is also complicated. There is an epimerase portion of the enzyme. There's a kinase portion of the enzyme. Most of the mutation in GNE are limited to the epimerase portion, but there are some mutations that also happen in the kinase portion. As I said, there is another disorder that happens with GNE mutation called sialidosis. That's primarily a pediatric disorder and does not look anything like GNE myopathy. The MRI is a very, very helpful tool. We use MRI all the time in clinical as well as a research setting. So early in the disease, you will see changes of stir signal. Stir signal is non-specific. It can happen from nerve damage. It can happen from muscle damage, but generally reflects some degree of swelling or edema in the muscle or inflammation in the muscle. And that's the white stuff that you see here in the hamstring muscles. These are the hamstring muscles. This is the quadriceps muscle. This is your femur bone. So this is the white stuff is the hamstring. Now, in our T1, which is mainly looking for fat, so you want your muscles to be gray like this. So that's again, tibia means that this is your calf muscles. This is your gastroc muscles on that. And then the white stuff is your tibialis anterior or shin muscles that have essentially turned into fat. There's no healthy muscle left there. This is your shin bone in the front. Whereas in the thigh or upper part, this is your femur bone, thigh bone. The quadriceps look fine, and that's why the quadriceps muscles are spared. But if you look at the hamstrings, behind the back of the thigh, most of it is fat, and that's the clinical picture we see as well. We see foot drop because of this, and we see weakness of the hamstrings because of this, but the quadriceps are strong, so your knee extension looks good. There are many mutations that cause this gene, but there are some patterns that we see. So there are specific mutations in the Chinese and the Japanese population. There are specific mutations in the Indian population, the V727M mutation. There's a mutation that originated from the Rajasthan region of India and somehow migrated to Europe and is seen in the Bulgarian Roma or the Gypsy population, but it's also quite common in India. And then there is the Middle Eastern population, the M743T. A lot of work is being done on that, and some of the gene therapy studies that you will see are primarily trying to attack this particular mutation. And then there is a pocket of GNE myopathy in Northern Europe, especially in Britain. I would have predicted that that's going to be seen primarily in the South Asians that are in England, but actually it's a Caucasian mutation, and they can present with GNE myopathy as well. So we talked about the sialic acid pathway, and the hope is that if you can somehow restore sialic acid production, it will eventually fix the disease, and that's what the holy grail in GNE myopathy is. The muscle is a very useful tool not only for diagnosis, but it's also a very helpful tool to look at disease progression, but also as an outcome measure for clinical trials. And MRI is being increasingly used in muscle research as an outcome measure. So if you can prevent this fat change in the muscles, that is a successful treatment. And these MRI changes correlate quite heavily with the muscle function testing that we do through a physical therapist. So there is high concordance between what's happening in the MRI, what's happening physically and functionally in the patients. So I'm going to switch gears now. I'm going to talk about clinical trials, and I'm not going to talk about stuff that was done in the 80s and 90s that was primarily looking at IVIG. And the reason they used IVIG because a lot of the IVIG molecules have a lot of sialic acid. So those trials were done at the NIH by Dr. Delakas and Dr. Plotz. Unfortunately, none of those treatments worked. The Ultragenics back in the late 2010s started a trial. This was a pivotal trial, so this is a phase 3 trial. I, as an investigator, and UCI as a site, participated in both the phase 2 and the phase 3 trial of extended release sialic acid, but we also participated in the natural history study, and we were the only site in the US. So patients were sent from all over the country to our site. And what's important, and I'm going to talk about this in my discussion about unmet need, is that your drug trial or your drug study is only as good as how carefully it is done, right? If a drug trial fails, it could be because the drug didn't work, or the drug trial failed because we were not careful about using the best outcome measure, or we were not careful in selecting the right patients on that. Ultragenics, to their credit, sent a physical therapist who spent 3 days with two of our physical therapists and really taught them how to carefully assess for changes in muscle strength and muscle function. And that was the beauty of this trial, is that the concordance between the two physical therapists and concordance between the physical therapist and other sites was very tight, which means that we really measured what was happening in the muscle. That's very important. Any future trial, you need to carefully train your evaluators, the therapists who are going to be doing the assessments. Otherwise, it's going to doom the trial, and that's very important. So this was a very carefully done trial. As you can see, they achieved their goal, which was to increase the amount of free sialic acid in the serum. So compared to the placebo arm, which is this line, the sialic acid did increase substantially and stayed increased throughout the trial. Despite that, unfortunately, there was no change in the upper extremity score, or did not reach statistical significance, and there was no change in the lower extremity composite score. And the lower extremity was problematic because the lower extremities get worse faster. So at some point, it's very difficult to measure the lower extremity composite score, whereas upper extremity still is a viable measure even in advanced disease. And that's why some of the new trials are primarily focusing on upper extremity score more than the lower extremity score. In this particular study, we also used dynamometry to look at knee extensor. This was a neat setup where you had a computer-based muscle testing score. And as you can see, the functional activity score for the GNE myopathy were no different between placebo and the active drug. So, and the same thing is seen some of the other measures, sit to stand test, the 30-second weighted arm lift test was the same. The 6-minute walk test was not much different. In fact, if anything, it actually declined a little bit more in the treatment arm on that. So none of these turned out to be significant, and we reported this as a negative trial in neurology in 2019. Now, after that trial finished, the group at NIH with Dr. Gall and Dr. Carrillo was working very heavily on this another compound that acted higher up in the pathway, enzyme pathway for sialic acid. It's a rate limiting enzyme called MANAC. And they had been doing some pivotal studies. So these were the phase 2 studies in GNE myopathy done at the NIH. These were, again, primarily Caucasian patients, very similar distribution to what we had seen in our phase 3 trial, with significant disability. They had low plasma sialic acid levels, etc. And they initially did a small phase 2 study with 12 patients, randomizing them to either 3-gram dose or 6-gram dose of Manac, and followed them in the long run on that. And they were able to show in this small study significant improvements in muscle outcome measures. Let me take you through that. So again, one of the measures that they were looking at was this lectin level. So looking at muscle and see if you can see some lectins that normally are dependent on sialic acid on the muscle membrane. So in an untreated patient, you don't see much lectin, but you can see lectin present in patients who were treated, and that's the quantitative measure of how much lectin you produce in the muscle. So it turns out to be a good biomarker for the study, and that's the biomarker that was also used in the phase 3 study as well. So in their study, with this small study of 12 patients, they showed saw meaningful differences between the placebo arm and the Manac arm. Oh, sorry, the natural history arm and the Manac arm. So they didn't have a placebo, but they compared it to what the natural history of the patient was. So they saw differences in the lower extremity strength, in the upper extremity strength, in the 6-minute walk distance, and a functional rating score. They used a different functional rating score other than GNE myopathy functional activity score, and they saw a difference. So based on this, there was a large trial, phase 3 trial that was designed. It was funded by the NIH, by 2 different institutes in the NIH. It was run through the NeuroNext consortium. NeuroNext consortium is a consortium that was designed to run efficient trials in neuroscience and muscle disorders. They mainly do phase 2 studies, but this was actually a phase 3 study. So if the drug had worked, you could have gone straight to the FDA to ask for permission to use this drug in clinical use. There are 25 centers in the NeuroNext consortium, but these centers had the patients, so they decided to participate in it. They successfully enrolled patients in this study. They didn't have a challenge with getting patients, but unfortunately, Manac did not show any benefit in this study. The study results have not been published yet. They have not been posted on the clinicaltrials.gov site. There was a press release issued by NeuroNext, but you can if you go to the Lydian Bioscience, and Lydian was the company that provided the Manac. They don't even mention Manac anymore. They have stopped development of this work, suggesting that it was a complete failure in terms of showing any benefit. Otherwise, usually pharmaceutical companies don't give up that easily. So this, unfortunately, despite the best intents and a really well done trial, this was not successful. And again, as I mentioned, This could be because of study design, this could be because of selection of wrong patients who are too advanced, have too much fatty replacement of the muscles, or it's just that the chemical doesn't work, and that's what we can devise. Now, in the last few years, there has been a lot of excitement with another agent, which ultimately leads to increase sialic acid. So this is the work from Dr. Shin in Korea. And this is mouse model. So this is looking at the GNE myopathy mouse models, which has a myopathy. And they gave this supplement called 6-sialylactose to these patients and showed that it does increase not only sialic acid, but also manac levels as well as 6-sialylactose levels. This is the sialic acid changes in there, and they were able to show that in at least in the mice, they were able to reverse the myopathic features in the mice, and this was seen at multiple levels, not only in terms of function in the mice, but also looking at the muscle pathology. They really were able to change some of the muscle pathology changes and really reduce the myopathic amount. They reduce the number of rimmed vacuoles. They reduce the amount of fibers with inclusion, and even some of the amyloid changes in the muscle. So their conclusion was that this may be potentially a treatment that should be tried in humans. And to go along with that, they devised a small trial that, again, we have a total of 20 patients. About 14 of them are in the active arm of the study and 6 of them in the placebo. All of this data is published and available in the public domain on that. But they were able to show improvement in the serum sialic acid levels, the free sialic acid levels. They were able to measure sialic acid in the red blood cells, which is a very important advancement. We can, rather than doing a muscle biopsy, you can actually look at the blood cells and not have to do a muscle biopsy. And they ultimately showed that there were improvement in the muscle strength. So this is looking at shoulder abduction, ability to carry, lift your arm, ability to flex your elbow. There was not as much change in the grip strength. Again, the lower extremity is problematic because those changes happen really fast. So the upper extremity composite score is much more reliable than the lower extremity composite score in GNE myopathy. And as you will see when I talk about the drug trials, that's the measure that's primarily going to be used for the new ultragenic study as well. The Japanese, the Japanese population tends to behave differently than most other populations in terms of how they respond to drug. So it's imperative that any drug trial that is done in the West needs to be repeated independently in Japan, and they decided to do a trial in Japan. And this is the extended release sialic acid trial in Japan. Again, it's the same drug that we had used back in 2018, which had failed to show any benefit in the US. The Japanese tried it again. And again, it was a small study. There's only 20 patients that were treated with this. This is the demographics of that. This is what they look like. This is what their baseline 6-minute walk distance was. This was their baseline disease duration in terms of years, the sit to stand test, the arm lift test, etc., and the sialic acid. There were no differences between the groups, but they were able to show production of sialic acid, increase in sialic acid levels in the system, and they were able to show a significant slowing down of the decline in muscle function and muscle strength, including sit to stand test, etc. So this was a positive result. They showed that there were disease progression was slower in patients who were taking the 6-gram dose compared to patients who were not taking it or taking the lower dose. And based on this small study of 20 patients with only 15 patients in the active trial arm, the Japanese government approved the use of sialic acid in clinical purpose. on that. It again, in the US, I'm not sure that our FDA would have allowed that, and I doubt very much that the European regulators would have allowed this. But in Japan, they were able to approve it. The challenge is, and I get asked this question all the time, is can we get the extended release sialic acid from Japan? And the answer is no. I've asked this question to my Japanese colleagues multiple times. It turns out that they can't even produce enough sialic acid for their own population. And at this point, to my understanding, there is no way to get sialic acid, extended release sialic acid from Japan. Not to say that the patients have not asked and the patients haven't tried. So this brings me to the new trial. So this is a newer formulation of sialic acid by Ultragenyx. It's a prodrug, and it's conjugated to a C16 carbon, and it ultimately in the body will result in production of sialic acid or release of sialic acid at a larger number. So this is a phase 1, phase 2. Phase 1 means that it's a first in human. So we need to make sure that it's safe. We need to make sure it's tolerable in humans. This will be a double blind, which means neither the patient or the physician that's administering the trial will know which drug assignment the patients will get. There is a placebo, which means that some of the patients may be getting placebo on that. And this is a ascending dose study, which means you start with a smaller dose and we generally go to a higher dose in the study. There are a total of 24 subjects that are planned to be randomized. There are going to be 2 cohorts of 12 subjects each. All of this information is in the public domain. These are taken from clinicaltrials.gov site. Initially, Ultragenyx had planned for 2 to 4 sites. In the end, there are only 2 sites. There's one on the West Coast, which is UC Irvine, and there's one on the East Coast, which is in Iselin, New Jersey. These are the contact information. So if you guys are interested in participating in this trial, this is the number and this is the email that you will need to go to. So just a little bit about the inclusion criteria. This, you have to have a previously confirmed genetic diagnosis of GNE myopathy. They are not doing any new genetic testing in this study, so we need to have a copy of the genetic testing results as part of the screening on that. And it has to be done from a CLIA certified lab. It cannot be a fly-by-night genetic testing laboratory. It has to be a clinically reliable testing. The other criteria is the patient has to be able to walk a minimum of 20 meters independently during screening. We do allow use of assistive devices and braces as part of the study. The other big importance is that you have normal bicep strength with some caveats in it. This will be assessed by a physical therapist using a handheld dynamometer. And so because that's one of the big measurements as well. So that is a criteria that's going to be in force during the trial. Again, being willing and able to comply with all the study procedures is an important requirement. There are going to be needle muscle biopsies. We've been doing a lot of needle biopsies. I personally have done about 150 needle biopsies over the last 2 to 3 years. It takes 10 minutes. It's minimally invasive, minimally painful, but there is going to be a needle biopsy in this study, and the patient has to be willing to go through a contraception because we don't know what the effects of these drugs are on fetuses and pregnancy. So if you're participating in this study, you have to agree to use a highly effective contraception, and that applies for males as well as the male partner. Again, any other experimental drug is not allowed. So if you happen to be taking supplements that you bought from the internet, whether it's Manac, whether it's extended release sialic acid, whether it's 6-allolactose, if you're taking intravenous immunoglobulins or any other supplements that may in the body turn into sialic acid, that has to be stopped at least 60 days prior to the study. Any changes in the diet or exercise routine will not be allowed in the 30 days prior to the study. So again, it's important to plan it accordingly. And then there are certain medications that have an effect on the enzymes that break up the drug will be not allowed. And if you're known to be allergic to acylic acid, obviously that will limit your participation in this study. And again, there are other biochemical features, including liver enzymes, kidney functions, etc., any heart conditions that will be an exclusion. And pregnancy or wanting to become pregnant is going to be an exclusion in this study. So there are only 24 patients. It's a 1 year study because the assessments are going to be at baseline and then what happens at 48 weeks. So it's primarily a safety and tolerability study. So our main interest is if there are treatment-related, treatment-emergent adverse events that happen with this study. But we are looking at upper extremity composite score. We're looking at free and total sialic acid levels in the muscle, again, change from baseline to 12 weeks. So there will be 2 biopsies at the minimum, baseline and 12 weeks. Lower extremity composite score, a 6-minute walk test, and a GNE myopathy functional activity score is going to be done on all. And all of these will be done by physical therapists who's going to be highly trained and certified to be able to do these measurements. There are you'll get assigned to either a low dose, which you'll continue for 48 weeks, and then you'll go into a long-term extension, or you'll be on a high dose for 48 weeks and then go into an extension phase of the study. And then if you happen to be in the placebo is linked to either a low dose or high dose. So at the end of the 48 weeks, you will be switched over to either a low dose or high dose, depending on which cohort you are assigned to. It's a 3-to-1 randomization, which means it really favors the patient. So you only have a 25% chance of being in the placebo arm, but you may still get assigned to placebo arm. And once you get assigned, you will stay in that placebo arm for the 48 weeks. So those are the current clinical trials. There are a significant number of unmet needs in GNE myopathy. There are things that we know, and there are things that we don't know. One of the biggest challenge in GNE myopathy is even though there have been multiple animal models that have been described, they tend to lose their phenotype. So even if they had muscle disease with the initial manifestation, with multiple generations or down the generations, they lose their muscle phenotype and they primarily have a kidney phenotype. We don't have good antibodies to GNE protein, which has become a big limitation in terms of doing experimental work, and a lot of people are working on that. There has been doubt raised about whether silation or the presence of sialic acid in muscle membrane is the causative mechanism. There's still debate about that. I'm a believer in sialic acid being the responsible pathology, so it's less of a concern to me. We don't have good reliable biomarkers. I think the work in the last 5 years has shown that we can use MRI as a reliable biomarker to monitor disease progression. We can use MRI in clinical trials to look at treatment outcomes. The lectin staining is a good measure. Measuring the sialic acid on red blood cells is a good measure, or the good old-fashioned of measuring sialic acid in muscle is also a good measure. There are a number of mutations. There are well over 100 mutations in GNE. Not all of them are necessarily disease-causing. Not all of them cause the disease to the same extent. So we still need some work on relying this. And we need better assays to measure GNE enzymatic functions. So just a quick few words. As I mentioned, Dr. Bhana, who's comes from the industry, he's the newest member of the board, was planning to do a detailed discussion on gene therapies at a future date. He wasn't able to join us today. So I look forward to his talk. I'm just going to briefly tell you about 3 gene therapy efforts that are in process. Now, it's a little sad that there are three competing efforts. I hope we combine our forces and do it as one force, but there is political challenges and unfortunate that we are spinning the wheels in so many different directions on that. But the one that I primarily am involved with is the effort by the Neuromuscular Disease Foundation. I am the clinical PI for this trial. Most of the preclinical work is being done right now through either contract research organizations. So these are non-academic labs that do this for a fee. So Charles River, which is a very reliable contract research lab, is doing a lot of the animal model generation, etc. And then you have scientists who have been funded by the Neuromuscular Disease Foundation, specifically Dr. Monkel Leck, who's a wonderful scientist at Yale, and Dr. Stella Rosenbaum Mitrani in Israel are doing a lot of the work related to that. It is going to be a small trial whenever it comes to clinical studies. This will be a virus-based or AAV virus, and AAV virus is the common virus that is being used in most of the gene therapy. We know a lot about it. We know how to mitigate the issues with it. But this is going to be a new generation or next generation AAV that is highly specific to the muscle. So that avoids the issue of using very high doses of the virus and should reduce some of the toxicity related to high dose of the virus use. Will address not only GNE function in the muscle, but it also will affect other non or extramuscular functions of the muscle. And again, the goal is to stabilize muscle degeneration. This is not likely to reverse muscle degeneration. That is a very, very high bar to achieve in disease. In neurology and muscle diseases, what we are hoping for at the minimum is to stopping the progression of the disease or significantly slowing down the progression of the disease. So that's what this trial is planning to do. So this is in a nutshell the people who are involved. So Monkel's lab in Yale, Dr. Mitrani Rosenbaum's lab in Hadassah are involved. We are working with commercial research organizations like Charles River Laboratory, and we have employed some regulatory experts and translational experts. And then there's a lot of assessment work that's being done by Marianne Husing at the NIH as part of that. So a initial pre-IND submission meeting was held with the NIH, and we are trying to work towards creating an IND. IND is the ultimate step where we tell the FDA that we are planning to do a study in humans on that. So the goal here is to deliver the complete gene through a AAV virus. This is a modification of an AAV virus called MyoAAV or myotrophic. Myotrophic means that it has a predilection to go to the muscle, so you can get away with much smaller doses of the virus than what you would normally require. And then there is a muscle promoter. So this is the muscle CK that's going to make sure that it not only gets into the muscle and gets activated in the muscle. It doesn't become permanently part of your DNA. So that's the beauty of AAV. It creates a little niche for itself in the cell, but it doesn't become permanent part of the muscle on that. And that's one of the advantages. So this is some of the data. It's a little too technical to talk about in a webinar, but there is clear production of GNE that happens in the muscle, and some of the preclinical work is definitely showing some benefit in that. And then you can look at muscle and kidney silation level in response to the treatment. The animal model that they're using doesn't have a very strong muscle phenotype. The animal model that Paul Martin probably has a better phenotype. So at some point we'd like to collaborate with Paul's model, but again, he works with Genocera. That's a competing organization. So there are political challenges that are going on there. And then this is some of the silation material in muscle and kidney. So the gene therapy does do what it's supposed to do. It delivers Again, we'd like to test it in other mouse models as well before we go to humans. And again, as I mentioned, one of the challenges in GNE is we don't have the greatest mouse models in GNE. So other gene therapy efforts, as I mentioned, includes Genocera. So Genocera is a biotech company that was spun off by Dr. Paul Martin, who's a researcher at Nationwide Children's Hospital. Again, it is another AAV vector-based gene therapy. They are planning to do a double gene delivery, and it'll be interesting to see if FDA will allow two different genes to be delivered on that. But what they're hoping to do is to do a GNE complete GNE gene that will correct the genetic defect. And then in addition, they want to deliver the gene for follistatin, which inhibits the myostatin, which is a muscle breakdown protein. So the hope here is that you can stop progression, but you can also improve muscle strength through follistatin inhibition. And the third effort is through Solve GNE, which is a consortium created by a group of GNE patients from Long Island and New York, working with Stanford, with John Day, and some of the other experts in Stanford. They have a wonderful website. They actually have a media that talks about a lot of the efforts that they are putting together. And this is a very interesting approach where they, through a lipoprotein or lipid nanoparticle production, they are hoping to deliver not only the correct GNE or the wild type GNE gene, but they also want to put in a short hair RNA inhibitor that can inhibit the dysfunctional gene. So this is primarily for the Middle Eastern GNE mutation. So the idea here would be not only are you delivering the correct gene, but you're also shutting down the expression of the mutant gene, and that should result in improvement. There's a there's a paper that just came out in Future Science Open Access, so everyone can have access to this. Very technically detailed and heavy in that. But the idea here is that you correct the gene defect by delivering the wild type gene, but you also knock down the expression of the mutant gene that results in normal sialic acid expression and that. So they've only done it in animal models so far. I think they are raising money and they are hoping to go to a clinical trial. So I'm going to stop here. A shameless plug for our neuromuscular colloquium, which is a wonderful national international meeting. It's going to happen next week on Huntington Beach. And we have a great group of pathologists and clinicians from all over the world who are participating in it. Hope to see some of you there. Thank you so much, and I'm happy to take any questions or comments.
We have, thank you very much, Dr. Mozaffar. That was excellent. We have several questions from the audience, so let's just jump right into them. People are asking, we have a couple questions about 6SL, whether it's effective or not for patients to take, despite the kind of murky clinical trial.
So again, it's only been used in one ethnic group, which is Koreans. So we don't know if it's going to work in other, because there are pharmacological differences on how body metabolizes stuff. It was a very small study, as I mentioned, it's only about 14 patients. And there was a small effect, but whether that will pan out in a larger study, I mean, remember what happened with sialic acid, extended-release sialic acid in the US, the phase 2 study with Ultragenyx had shown benefit. It did not pan out in phase 3. Manac showed a benefit in phase 2 that did not pan out in a larger study. So you have to be really careful and conservative interpreting small studies. There's a reason why you have to have the right sample size calculation. So it really needs to be done as a larger study. I was involved with the Treat NMD Therapeutic, the TACT committee, and they recently met with the Korean investigators, advising them on whether further studies need to be done or not. They are interested in doing a larger study, but I think from a financial limitation, they would like to restrict it to one or two countries. So it's not really clear what their ultimate plan would be. I think they were looking at doing it either in Korea and India or Korea or China as their next phase.
Interesting. Here's another question. We have two questions about the extended release sialic acid trials. Obviously, one, Japan has approved this, and they had significant difference in their trial, whereas the Ultragenyx study didn't meet its primary endpoints. The questions are about whether this was whether the difference might be a clinical trial design issue. And there's a secondary question about whether there were any subgroup analyses based on gene variant that might account for the differences between the studies.
So, I mean, again, the Japanese trial was really, really small. I would compare it to the phase two of the extended release salic acid that Ultragenyx had done that had shown a positive signal. I would compare it to the phase two of the MANAC trial that was done and had shown a positive thing. 15 patients or 20 patients are not enough to come to meaningful conclusions on that. And we definitely won't have enough strength to look at mutation differences as an explanation for this. As I said, there is a positive data, but it's again, my enthusiasm is reduced by the small number of patients that were done in this study. And I said most regulatory agencies, including the FDA or the EMA, which is the European, would not have accepted that result for drug approval. So the drug got approved in Japan. Part of it may be pressure or emotional decision, but the scientific data is not strong enough to say that this drug really works, and we just definitely don't have enough numbers there to make meaningful conclusion whether it works in certain mutations versus other mutations.
Okay. Here's another one for you. Have the natural history studies shown any component of coexistent peripheral or small fiber neuropathy in genome?
To my knowledge, no. But I don't think that was the focus of the natural history studies. The patients usually don't complain of neuropathy. I mean, again, when somebody presents with foot drop, the natural expectation is that this has to be nerve related, especially if they don't have a family history of GNE myopathy. The obvious bias is that this has to be coming from the nerves or coming from the spine. And I've been guilty of that. My first two patients that I ever saw with GNE myopathy, I was convinced that they had a nerve dysfunction. There has been no evidence on nerve conductions, on muscle biopsies, that there is a significant neuropathy or peripheral nerve component to it. Small fiber neuropathy causes pain, it causes nuisance, but it doesn't cause functional loss.
Okay. Here's another one for regarding the Ultragenyx study. There's been recent FDA guidance for rare disease studies that may allow trial designs without a placebo group, instead using natural history for comparison. Why didn't Ultragenyx choose this route to spare patients from taking placebo?
So again, how much of that is in public domain, I don't know. They do have a good explanation in their study protocol and why they chose to do it this way. That's probably a better question to ask Ultragenyx. Again, yes, there are natural history studies. They are very well natural history, but natural history is not the same as doing a placebo group. There is a definite placebo effect that you don't get from natural history study. So I personally am not a big fan of doing a comparator with a natural history arm as opposed to doing an actual placebo. They have minimized the number of placebo patients, so it's a 3-to-1 randomization, which is exceedingly favorable to patient, but it will pick up that small placebo effect. In neuromuscular diseases, you may have placebo effect as much as 40%. We've learned that from myasthenia and other studies. So I personally would not be brave enough to do a trial without a placebo in an exceedingly complicated disease like this.
That makes sense. Here's another one. Why haven't anabolic steroids been tried as a treatment or peptides? Very popular now, these peptides.
I'm sorry, can you repeat that question, Colin?
Uh, Someone's asking whether anabolic steroids have been considered as a potential treatment, and they also are asking about peptides.
So, I mean, anabolic steroids have been tried in all sorts of muscle diseases, right? So, the problem is anabolic steroids do increase muscle mass. Same thing with myostatin inhibitors. I mean, I'm not the greatest fan of myostatin inhibitors, and that's why I'm not as enthused about the the Genosera approach, because myostatin or steroids work in normal muscle. So, if you have a healthy athlete and you give them anabolic steroids, or if you have a healthy athlete and you give them a myostatin inhibitor, their muscle will get bigger, usually about 20% increase in muscle mass, and that muscle mass increase will result in a functional improvement or strength improvement in the muscle. Now, if you have a muscle condition where there is ongoing consistent damage to the muscle, none of these effects show. People have done anabolic steroid trials in Duchenne muscular dystrophy and other forms of muscular dystrophy. There have been a number of trials on the myostatin pathway in FSH and Duchenne, in Charcot-Marie-Tooth disease. Yes, there is an increase in muscle mass, but it does not translate into muscle strength improvement. Anabolic steroids have challenges. They are toxic to the liver. They promote um, prostate enlargement or prostate cancer. So they are definitely not benign. They cause virilization, which they make people manly, and especially in women, that's not a very desirable side effect.
Here's another question. Could a bispecific muscle-targeted siRNA conjugate with a muscle-homing peptide therapy be a viable treatment? Similar research and clinical trials are being carried out for FSHD1 and DM1 by Sarepta.
So that is the approach that Solve GNE is thinking about, and they're working with a company called Neuromanides. Their approach would be to deliver a normal GNE gene, but also a siRNA to suppress the mutant gene on that. So that's the approach they are taking for a bispecific. The approach that Genosera is taking is they want to deliver a normal gene, but they also want to deliver a follistatin gene, which is fine, but I just gave you the rationale why follistatin is not the greatest choice in a disease muscle. If this was a normal athlete and I want to do gene doping so they can win the Olympics, great idea. I don't think it works in disease animals. The only drug to that effect that has shown that is the Scholar Rock drug, where they have shown some benefit in SMA. But again, generally, patients with ongoing disease in the muscle or nerve do not respond as well to myostatin inhibitors.
Okay. Another question we have, we have someone who'd like you to just explain what a prodrug is generally.
So a prodrug is a drug that is in a different form or a different conjugate, and once it goes into the body, it gets converted to the actual drug. So it's like using a Trojan horse to deliver a drug, and in the body, then it gets changed to the real drug that you really need. Very simple explanation, very simplistic explanation, but that's what it is.
Excellent.
And I think Dr. Angela Leck is making a comment about this a difference between a dominant disease versus a recessive disease. So, FSHD and myotonic dystrophy type 1 are examples of dominant disease where you have one gene that is causing a toxic effect. So, if you shut down that gene, you can fix the problem. The problem with recessive disease, such as GNE myopathy, is that you have two genes, two copies of the gene that are causing the problem. So knocking out one copy will not do the trick. You really require a healthy copy of the gene.
I see. Another question, how about colostrum supplements, which are supposedly contain sialic acid? Has that been considered as
I really have no knowledge of this. I would not know this answer.
understood? Um, Here's another one about the Ultragenics trial. What is your personal outlook on the Ultragenics trial based on your scientific knowledge compared to the prior failed trials?
Well, I mean, again, I am hopeful. I mean, so if I was not a believer in the sialic acid pathway, I would have been skeptical about it. I still think sialic acid pathway is relevant and valid. It's just a matter of delivering a drug that can produce right amount of sialic acid and using a appropriate types of patient who don't have too advanced of a disease, who don't have too much fat replacement, has still have viable muscles, and using measurements that are sensitive to disease change in response to a treatment and be done in a valid, reliable way. So again, we are very excited for this study. We are gearing up to do this study. It's going to be a challenge because we will be only working with 24 patients across two sites. So technically 12 patients per site. And we will have a lot of patients who might get unhappy with us because we were not able to accommodate them in the study.
Here's another question. Someone's asking about the variants in gene mutations that cause the disease, and they're asking when we refer to the Middle East strain, what does that cover any other regions, or is that restricted to the groups that you mentioned?
So the Middle Eastern strain is the strain that was primarily seen in the Persian Jewish population, but is also present in the non-Jewish Persian populations and also in some of the Middle Eastern, like in the Arabian Peninsula as well. It is a well-known mutation. We know a lot about how that behaves in the clinical setting, what the rate of progression is, etc. But that's not only the only mutation. There are other mutations. There are two mutations in India and South Asia. There's a mutation in Japan. So there are multiple mutations that cause the disease. So the Middle Eastern mutation, because in the US, GNE is a bicoastal disease primarily. So we see a large population set in New York. We see even larger population set in West LA or the thing. And then there's a pocket in West Vancouver that most of them are have the Middle Eastern mutation, but there's plenty. The example I quote is I'm about 50 miles south of UCLA. And most of the patients at UCLA are Persian Jewish with the Middle Eastern mutation. 50 miles south in Irvine, majority of my patients are neither Persian or Jewish. And most of them have other mutations other than the Middle Eastern mutation.
Okay. Yeah, Dr. Leck is also adding in the Q&A that this M743T DNA change is common in the Middle Eastern population, but it doesn't mean people outside that region couldn't have this mutation as well. Um,
The more you look for it, the more you'll find the GNE mutation. I think
yes.
that we are starting to realize that it is one of the more common reasons for a limb-girdle pattern muscle weakness.
Here's another question. What is C16's involvement in the UX016 trial? Is it common, safe, and or tested in other treatments?
So C16 is just think of it as a Trojan horse. I mean, it's conjugated to C16 to enter the body and then get delivered as the silic acid. So it's a prodrug conjugation that's happening. So it's combining with C16, which is lipid rich, so it enters the muscle more easily. But think of it as purely as a Trojan horse. And one of the reasons to doing the study is to make sure that giving a drug that's conjugated with C16 is safe and tolerable.
I think that's almost all the questions. We have one more though, because this is, when will the Ultragenics trial start?
We're almost there. I think our regulatory stuff is in its final segment. I would say probably 1 to 2 months. We have a designated coordinator for it, so she will start reaching out to people on the list that we have created on that. So I would say probably in the next 1 to 2 months is when we start reaching out to patients.
Great, thank you so much. I think that's it for our questions. If anyone has anything else, fire away now. Well, I
Coleman,
guess
this is Tara.
hi.
If any patients in our community have additional questions, would you please send them to info@curegnem.org, and we will attempt to answer your questions.
That's great. Yes, we hope everyone found today's webinar informative. This webinar is recorded and it'll be available on our website and our YouTube channel. As Dr. Mozaffar mentioned, we will also be hosting in the next weeks to months a webinar focused on our gene therapy program. That's progressed significantly over the past year. We recently had feedback from the FDA on the additional work we need to perform to move ahead into the clinic. We're working hard on these steps right now, thanks to generous donors, but we're going to need significantly more funding to reach the clinic. So if you are interested in learning more about NDF or supporting our research and advocacy efforts, please get in touch. That email that Tara just mentioned, info@curegnem.org. And I think that's it. Thank you all very much for joining us.
Thank you.
Thank you, Dr. Mozaffar.